Hello class! This is me again, Sir Lars. Oh, remember me? Baka nagka-amnesia na agad kayo. So again, I am the one who handled your basic organic chemistry. And here, in this video, I am going to start our discussion on your biochemistry crash courses. Class, again, remember, I will handle all your crash courses 1 to 10. And here, in our slides is your detailed schedule for your biochem crash course 1 to 10. Alright, 1A. Again, if you're in this video, I hope that you have already watched our cue point for your organic chem and your biochemistry before diving into this video. Kasi baka magulat kayo because, of course, very different ang nature of our biochem discussion. So class, anyway, again, for our biochem, we will tackle your biomolecules, your cellular respiration, and your metabolic pathways, particularly si carbs, lipids, and amino acids. And finally, of course, we also have the discussion on central dogma and your characterization in biochem lab techniques. So class, please see your updated schedule for your biochemistry crash courses. Okay? So again, for our biochemistry crash course, we will start with our brief intro. All ready na? If you are ready, then I will proceed in discussing our introduction to biochem. So class, for our first topic, okay, this will be just a brief introduction on biochem. So here, we are going to cover brief overview, your cells, particularly its structure and functions. And lastly, water and its properties. So guys, take note for our discussion on water. Baka mag-expect kayo na this will be very lengthy. So we will just cover those relevant to our discussion in biochem. Okay? So before I formally start my discussion, I prepared a mini-game. So familiar ba with 4 picks 1 word? So this will be called as your quick quest, o, diba? So for our first item here in our Q quest, ayan, so we have 4 pictures. Okay, and your task is to identify the common theme of these 4 pictures. Class, you can pause the video so that you can guess the answer. So I will give the answer. So first pala, ayan, may clue ako. Okay, second word is I, third word is F. So actually class, so if you will see, if you will analyze the pictures, we have here, the first picture is your DNA, the blueprint of life. Okay? So here we have your cells, the fundamental units of life. Lahat yan class, it depicts your life. Sir, ano yung third picture? So this is not related to our discussion, but this is an electrocardiogram monitoring your heartbeat. Okay? And our fourth, ayan, fourth picture is a picture of your chloroplast, your plant cell. Okay, so for our next item, ayan, so very easy lang to class. Okay, so here you can see a water molecule. Here, for our third and fourth picture, this is the ball and stick model of your molecules, inorganic molecules and organic molecules. And here, our second picture is a picture of your specific enzyme. So here, our answer is, ayan, molecule, diba? So last class, I have last item here. So this is the pictures. Ayan, so here you can see that these are all, what's your answer? Tubig. Sir, pwede po ba yung water? Of course, ayan. So here, these 3 concepts or themes that we displayed in our mini icebreaker are related in our discussion in our overview. All right, class, to formally start our discussion, we know that biochem is the study of life at the molecular level. So here in our discussion, we will focus on our 4 biomolecules and the processes that they participate to. Class, check your manual. So there in your biochem notes, we gave you a concept map that summarizes the discussion that we will cover in our biochem discussions of crash course. Class, for our first part of discussion, here is our topic, the 4 biomolecules. So it involves carbs, lipids, proteins, and nucleic acids. And remember, our focus here are their structure, function, and properties. Okay, so afterwards, we will also tackle the respective processes. So we have your metabolic pathways of carbohydrates, your lipids, and amino acid. Okay, but of course, important in class that we tackle your cellular respiration because a big chunk, okay? So many items appear in the board exam is all about your cellular respiration. All right, so for nucleic acids naman, we will cover your central dogma. Sir, are we going to cover as well the laboratory techniques? Yes, okay? So it appears din kasi class sa biochem subtest ng chem board exam. Class, of course, expect. Ayan. So, ang biochem kasi, that's just an applied organic chemistry in biology. So, every now and then, on our discussion, I'm going, okay, to review, to have a recall of some organic chemistry core concepts for you to better understand our discussion here in biochem. Okay, so now let's start with our cells. Okay, so as I have said earlier, cells are the fundamental units of life. So of course, these are further classified into 2 broad categories. So we have your prokaryotic cell and we also have your eukaryotic cells. So sa eukaryotic cells class, we have 2 subtypes. Familiar. So here at the left side is your animal cell. And for our right side here is your plant cell. Sir, may difference ba sa kanila? Yes. But for now, let's examine first your prokaryotic cell. Based on its structure, very simple si prokaryotic cell. As you can see class, wala siyang organelles. So yung DNA niya is in the cytosol. Of course, as well as your your ribosomes. Sir, wait lang, ano po ang difference ni cytosol and cytoplasm? Class, let me clarify. Si cytosol po is the fluid component. Okay? Si cytoplasm, that includes your small molecules and the nutrients in your cytosol. Okay? Again, si cytosol, fluid component, cytoplasm, fluid plus nutrients. Alright? So going back, let's examine naman your eukaryotic cells. So as you can see, much more complex ang structure ni eukaryotic cell. So for eukaryotic cells, both your animal cells and your plant cells, they have organelles. And these organelles have specific functions. So in your manual, we listed some relevant organelles that you need to familiarize. So later on, we will tackle more on that. But for now, let's focus first on cellular organization. O, diba? So ang cells class, its structure is very systematic. Organized po siya. It has a hierarchy. As stated here, cells are organized hierarchically. So starting from your atoms to your biomolecules in order to form your organelles. That's why very complex ang structure ni eukaryotes compared to your prokaryotes na may simple structure lang. So organelles are very important for specific functions of your cell. Ito class ang visualization ng organization of your cell. So this is the cell containing its organelles. And these organelles are made up of different supramolecular complexes. So for instance, sa nucleus, it has a chromatin component. All right, so this is an organelle. Si plasma membrane is also an organelle as well as the cell wall. And we treat them as your supramolecular complexes. And that's on your level 3. Sa level 2 naman, these are your macromolecules, or here in biochem, we call them your biomolecules. Check. Your chromatin is made up of DNA. Your plasma membrane, one of its components are your proteins. And for cell wall, its main component is cellulose. And come your level 1, we can see that they are the monomeric units of your biomolecules. O class ha, I expect that somehow you have knowledge on this monomeric units from your undergrad biochem. So for DNA, monomeric unit niya po, nucleic acid. For protein, we have amino acid. And for cellulose, we have carbohydrate. Alright, so class, I have here some images of your relevant organelles. So feel free to guess or name these organelles. Okay, so number 1, so what is this organelle? So that is your nucleus. Okay, so number 2, sir, ano yan? Is that an enzyme? No po. So this is actually a visualization of your ribosome with its 2 subunits. Later on, we will discuss this on your central dogma. So much relevant or more relevant siya sa discussion on central dogma. Third, di ba ang powerhouse ng cell, that is your mitochondria. All right, so number 4, we have your chloroplast. Okay, number 5, I think you are already familiar. So this is your lipid bilayer, the structure of your plasma membrane. And lastly, number 6, so this is very important in post-processing in your central dogma. That is your ER, or your endoplasmic reticulum. Recall class, we have smooth and rough ER in our cells, inside our cells. So ang dali, diba? So of course, this is just an introduction to your cellular structures. Lastly class, here is the summary of your organelles. Again, very important po si organelles for cell survival. Ayan, emphasis on survival and activity as well as to maintain homeostasis. All right, so class, here listed in our table is are the relevant organelles that we will encounter in our discussion. So please read this in your manual. Reading assignment po yan for you. Okay, so that's it for our cells and its structure and function. So now let's proceed with our last subtopic for our intro to biochem. Ayan, so here we have water. Sir, why do we need to discuss water in biochem? So class, as we all know, major component po ng cytosol is water. Okay? So they are, your cytosol is in aqueous environment. All right? So let's enumerate the unique properties of water. Diba? So of course, from our high school chem, we know si water ay universal solvent because of its structure. So examine the structure of water. It has bend shape, or recall your inorganic chem discussion. And because of that, it is also highly polar, allowing up to 4 hydrogen bonds. So again, capable si water of forming 4 hydrogen bonds because of the lone pairs in its structure. Class, for your water, it has a partially negative region because of the lone pairs of electrons and a partially positive region in your hydrogen atom. Okay? That's why it is capable of interacting with polar and charged molecules. Okay? And lastly, si water po is very crucial in maintaining your acid base and buffering systems. For our biological systems class, we have natural buffer systems, for instance, bicarbonate. And this is very important in order for our biomolecules to be functionally active. So we will focus this on our discussion on enzymes. Okay, so sit back and relax muna kayo. So of course, lastly, because of these structural properties of water, water also has high surface tension and capillary action. So here in our GIF, it displays your high surface tension of water, allowing transport of organisms. All right. So class, because of these unique properties of water, we will also discuss the two important characteristics of your water, particularly cohesion and adhesion. Sir, what is the difference between these two functional properties? Ayan. So when we say cohesion, this is an attraction between water molecules. Okay, similar water molecules. So as you can see here in our picture, your cohesion is the attraction between water molecules. Si adhesion naman is the attraction between water and other surfaces. But class here, remember, si water, it has a certain or strong affinity with polar or charged surfaces. Okay, again, polar and charged surfaces lang po. All right, so for cohesion, ang main cause, strong hydrogen bonding among your water molecules. While for your adhesive property, it's the interaction, the strong interaction with your water and polar or charged surfaces. Okay, that's why si cohesion po, it gives rise to the high surface tension of water, while si adhesion, it enables capillary action. So si capillary action class, this is very relevant in transport in plants. All right, so of course, again, remember your cohesion, attraction between water, adhesion, attraction between water and polar or charged surfaces. Okay, so that's it for our brief intro to biochem. So now, we will move forward with our discussion on biomolecules. Class, recall from your undergrad biochem, we have four main biomolecules that is very relevant or very relevant in our discussion in biochem. First one, we have your nucleic acid, carbohydrate, lipid, and protein. So this is just an animation that showcase the different monomeric units of your four different biomolecules. So class, remember, among the four biomolecules, your lipid is the only one that is not polymeric. As early as now, I'm going to repeat it because this appeared on the 2023 and I guess 2024, last year's board exam. Okay, so now we will first tackle our carbohydrates biomolecule. All right, so class, before I forgot pala ulit, in your biochem notes, at the end of your nucleic acids, we have compiled there the important information or summary of the important properties of your biomolecules. Okay, so now going back with our discussion, we have carbohydrates. So for all biomolecules, our pattern of discussion will be first the structure, then the function, and then some special discussion depending on the nature of those biomolecules. So here for carbs, we have your monosaccharides. And then we will also tackle glycosidic band. This is very important because from last year, medyo naging challenging ang discussion on glycosidic band. And lastly, I'm sorry, second to the last, your disaccharides and polysaccharides. So letter A and letter C subtopics are your classifications of carbohydrates. And lastly, again, for carbohydrates, we have a special discussion on chemical characterization. Okay. All right, class. To formally define carbohydrates, okay, so based on its structure, they are classified as polyhydroxy, meaning it has numerous hydroxyl groups in its structure. So polyhydroxy aldehydes and or ketones. So depending on the functional group present in its structure, okay? So of course, it can be classified to simple or complex. So later on, we have a slide for this classification. But for now, here is the overview of your carbohydrates. So class, points here that you need to know is that their feature. So it is polymeric and chiral. So later on, we will see the chiral centers of your carbohydrates. Main classifications, we have your mono, di, tri, and or polysaccharide. Okay? And for bond types, so this is the specific bond that joins your monosaccharide units together. So we have your glycosidic bond. And later, I will ask you what is the nature or what functional group is present in this glycosidic bond. And lastly, ang key function ng carbohydrate is mainly for structural. And of course, in our meta pathways, you will realize that it also has an energy function. Okay, so now let's start our discussion on monosaccharides. So monosaccharides class, we also know this as your simple sugar, the building blocks of our carbohydrates. So of course, these monosaccharides, we can further classify them into two categories. All right, so one, depending on the length of carbon chain. And the second one is dependent on the functional group present in its structure. For length of carbon chain, ang relevant lang sa discussion natin starts with your triose, tetrose, pentose, and then hexose. So that's for your 3 to 6 carbon sugar. For functional group, it depends on the functional group present in a structure. It can be an aldose or a ketose. So let's examine the structure based on the functional group classification. Okay, so this is a typical example of your monosaccharide. Class, remember what type of model is this? So this is your Fisher projection model. And always for your monosaccharide, we put priority to the carbon that participates in the carbonyl formation. So here is the C1 position, C2, C3, C4, C5, and then the last carbon will be positioned at C6. Okay, so this is an example of your aldose molecule because of the presence of your aldehyde functional group. O sir, sabi mo earlier, your monosaccharides or in general carbohydrates are chiral. Nasaan po yung chiral center? So class, in the Fisher projection model, it's really easier to visualize the chiral center because they are just the intersection in your structure. So sir, meaning si C2 until C5, they are the chiral centers? Yes po. Or remember our definition of chiral centers, it must have 4 different functional groups attached to a single carbon. Okay, so of course, again, this specific example is an aldose in terms of its functional group. But of course, in terms of the length of carbon chain, as we have seen earlier, it has 6 carbons. That's why it is a hexose sugar. So combining these two classifications, we call this monosaccharide as an aldohexose. So yan pala si glucose? Yes po. Okay, very common example, si glucose as an aldohexose sugar. O class, if you can see or observe, lahat po ng carbohydrates natin, they end in the suffix OSE. All right. So moving forward, we also have a known example for your ketose functional group. O, diba? As stated here, pag ketose, the ketone group is positioned at C2 of the structure. All right. So as you can see, that's the ketone functional group. And of course, priority pa rin po ang numbering ng carbonyl carbon. All right. So this is actually your fructose, a keto hexose sugar. O sir, do we need to memorize the structure? Yes po, somehow. So later on, I will share a tip on how to memorize efficiently the pentoses and hexoses because it will really help you a lot if you know their structures. Okay, so class, this list sorry of your monosaccharides are already in your manual. So I will give this to you again as a reading assignment. All right, so now we proceed with your D versus L configuration. Sir, why? Why do we need to discuss D versus L? Because class, your sugars, I mean your carbohydrates, or in general, your biomolecules, their configuration affects their natural distribution. So later on, we will see that meron pong bias ang nature in terms of the localization or existence of our sugars. But now, let's just brush up your knowledge on the concept of D versus L configuration. All right, so here is your reference sugar molecule. That is your glyceraldehyde. So glyceraldehyde class is a chiral molecule. As you can see here, it has a 1 chiral center. Okay, so let's treat the broken line as the plane of mirror plane for this discussion. So here is the similar structure of your glyceraldehyde. O sir, ano po yung gray structure dyan? So that is the precursor molecule of your glyceraldehyde molecule. That's your glycerol, a triol compound. Let's see again that this is the chiral center of your glyceraldehyde molecule. So in Fisher projection, that is the intersection. And remember class, if a molecule is chiral, it has the ability to rotate plane polarized light through optical rotation experiments. Again, through optical rotation experiments or through polarimetry experiments. Okay, so from experiments, if your molecule rotates plane polarized light to clockwise position, we call them your dextrorotatory sugar. All right, so if it rotates plane polarized light at counterclockwise direction, of course, we call them levorotatory. So that's the observation in your polarimetry experiments. However, we can also categorize your sugar based on their configuration. Okay? So, yun na nga yung D versus L configuration. Sir, paano po ang difference ng D at L? So, we call your glyceraldehyde D kapag D yung farthest hydroxyl group is at the right side of your Fisher projection. So, later on, we will have other examples that will clearly illustrate this D configuration. And on the other hand, kapag L naman po, it's at the right side, yung hydroxyl group. All right, so class, this discussion is important because again, it affects the distribution of your biomolecules. For instance, class, for our monosaccharides, naturally occurring sugars po ay nasa D configuration. Okay, so aside from its effect in its natural distribution, of course, it also affects their chemical, physical, sensory, and nutritional value. Okay, so class, again, I would like to emphasize here that the dextro and levo optical rotation configuration cannot be defined or cannot be determined through the structure of your sugar molecule. Only the D versus L configuration. Okay, again, hindi po synonymous yan na si dextro and levo. This can only be determined through experiments. All right, so let's proceed with our relevant examples of monosaccharides. First one for triose, as we have seen earlier, we have your glyceraldehyde molecule. So this is the structure of your glyceraldehyde molecule. For tetros, we have two D isomers. Class, again, lahat ng examples that I will flash are all in B configuration. So for our tetros, we have erythrose and threose. So ayan class, here, much clear ang illustration ng D isomer natin. So as you can see here, the farthest hydroxyl group is always at the right for D isomers or D configuration. Okay? So class, please familiarize the structure of the relevant monosaccharide examples. Now we go to our pentose. Naku sir, parami yung examples. Of course, that's why I'm going to share a tip and a mnemonic. Alright, so that B pentoses, we have four. These are your ribose, arabinose, xylose, and lysose. Alright, and for our mnemonics, we have ribs are extra lean. So sir, how can we determine the structure of these four D pentoses? Okay, class. Again, for visualization of our monosaccharides, we use Fisher projection. And as you can see, we have a pattern here. So starting from triose, it has a 1 intersection. Triose is a 3-carbon sugar, and its intersection is only 1. Now, if it's a 4-carbon sugar, tetrose, nag-increase by 1 ang intersection or chiral center. Okay? So that means plus 1 lang po tayo ng plus 1 in terms of the intersection in your Fisher projection. So sir, meaning if 2 ang ang intersection ng tetroses, meaning kay pentoses, 3, yes. And that's your starting point in drawing the structure. So now, after drawing the structure with 3 intersections in your Fisher projection, you will put the aldehyde functional group at the top of the skeleton and the methyl alcohol at the end or bottom of the skeleton. And from that, as I have said earlier, lahat ng D-sugar nasa right side ang hydroxyl group at the farthest end, okay, or at the farthest hydroxyl. So meaning, you will draw already the hydroxyl group farthest to your aldehyde functional group at the right side. And from that, you can use my tip. So for pentoses, you will put the hydroxyl group attached directly to the nearby carbon or nearby carbon at the farthest hydroxyl group to the right. So here we have right, right, left, left. Okay? And then the next carbon will follow here, alternating na lang po siya. So 1 per 1 for hydroxyl group. So right, left, right, left. And now, you can determine the structure. Again, prioritize the D hydroxyl groups at the right side of all D pentoses. And then, next one is the carbon nearby that hydroxyl group. So right, right, left, left. And then lastly, the last carbon will follow, last chiral carbon. We have an alternating right, left, right, left. Okay, so I hope that's clear, class. So that will really help you in familiarizing the structure. So for our hexoses, ayan, class. So medyo challenging na. That's why I shared you the tip. So sir, meaning kaya rin po bang gamitin yung tip for our D-hexoses? Yes po. Alright, so for our mnemonics, we have all altruists gladly make gum in gallon tanks. Okay, so that's for your 8D hexoses. A similar strategy. So for pentoses, we have 3 intersection. For your hexoses, plus 1 lang po. We have 4 intersections, as you can see in the Fisher projection structures of your hexoses. And from that, again, prioritize drawing the hydroxyl group farthest to the aldehyde group at the right side, because again, they are the isomers. So here, we will put it at the right side. As you can see, class, colored in red in our slides. And from that, yung nearby carbon from that farthest hydroxyl group, you will just put 4 rights and 4 lefts for their hydroxyl group. See, class, right, 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 left, 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 left. Diba? May pattern na. So that's easier to draw their structure. Okay? So the next chiral carbon will follow. So if 4 R's, 4 L's earlier, now we will only have 2 R's and 2 L's. So right, right, left, left, right, right, left, left. And lastly, the last chiral carbon with respect to the farthest hydroxyl group will only have an alternating pattern. Right, left, right, left, right, left, right, left. O sir, ang dali lang palang mag-draw ng structure kapag may mnemonics and strategy. Yes po. Okay, so that's the simple technique in order to draw efficiently your pentoses and hexoses. So class, I am emphasizing this because this topic, your pentoses and hexoses, always appear in the board exam. And sayang naman yung point because straightforward yung question. Okay, so again, for your aldohexoses, only glucose and galactose are very common in nature. Okay, so glucose is the most abundant of all aldoses and it comes naturally from the hydrolysis of your starch and cellulose. So ito ang structure ng glucose. For galactose naman, it is from the hydrolysis of fruit pectins. All right, class, I still have an additional discussion for hexoses. Okay, so examine the structure of your hexoses, particularly glucose and mannose, and then the pair of glucose and galactose. As you can see in their structure, they only differ in one stereogenic center. Okay, meaning yung configuration in the stereogenic center, one is at the right and one hydroxyl group is at the left. Okay, and that relationship, we call them epimers. So they are diastereomers that only differ in one stereogenic center in terms of their configuration. Okay? So for glucose, again, we have two common examples. See the structure of glucose and mannose. They are only epimeric in C2 position. O sir, nasan yung C2 position? So this is the first carbon, and this is the C2 position. For glucose, si hydroxyl group is at the right side, mannose is at the left side. Okay, so for the second pair, we have glucose and galactose at C4 position. C4 position class is C1, C2, C3, C4. There you go. So as you can see again, for glucose, right side si hydroxyl, for galactose, left side po si hydroxyl group. Class, remember these two epimeric pairs as this always appears also in your board exam. Okay, so again, C2, glucose mannose, C4, glucose galactose. Okay, so class, before I move forward, examine also the reactive functional groups present in your monosaccharide. Okay, so pause the video, recall your ORChem. What are the two reactive functional groups present in your carbohydrates, particularly monosaccharides? Diba? First one, we have the carbonyl carbon participating in your ketone or aldehyde functional group. Very reactive yan, okay? Because of the presence of your very electronegative oxygen doubly banded to your carbon. All right? And the second one is your hydroxyl group. We have numerous hydroxyl group, but here, ang reactive po because of its availability is the methyl alcohol or hydroxyl functional group. Again, because of the electronegative oxygen. That's why ang monosaccharides, they are capable of intramolecular reaction. Particularly, your monosaccharides are capable of intramolecular cyclization. Again, that is due to the presence of hydroxyl and carbonyl group in its structure. So let's discuss in brief the arrow pushing mechanism. So here is the electronegative oxygen of the hydroxyl group, and it will attack the electron deficient carbonyl carbon. So its product will be called as hemiacetal. All right, so that's both for your hexose and ketose sugar. Aldose and ketose sugar, sorry. So your aldohexoses in particular will form a six-membered ring, and in contrast to your ketohexoses, they will form five-membered ring. So for aldohexoses, we call it a pyranose ring. Or recall kapag carbohydrate, we add OSE as the suffix. All right, so cyclization results in the formation of a new stereogenic center. Sir, may new character unlocked? Yes. So that is your anomeric carbon, which is the previously carbonyl carbon. Again, our product is a hemiacetal. So hemiacetal is formally defined as a carbon that is bonded to a hydroxyl group and an ether functional group. All right, so cyclization will give rise to two different anomeric isomers because again, cyclization is not stereo-specific. That's why you need to familiarize these two isomers. So kapag alpha, ang tip ko dyan, alpha baba because the hydroxyl group is below the plane of the ring, this one. For your beta, of course, the opposite is true, beta-as. Nasa above ng plane si hydroxyl group. Alright, and these are the three structures of your monosaccharide, particularly glucose. Sir, do they coexist in dissolution? Yes. And that coexistence, we call them mutarotation. Alright, and upon reaching equilibrium, may preference po ang three structures na to. O ayan, at equilibrium, 37%, they prefer the alpha anomeric form. Alright, and then 63%, they prefer the beta anomeric form. Okay, and then trace amounts, it only have, it prefers the acyclic hydroxyaldehyde. So class, in terms of stability kasi, is a cyclic ring structure are more stable because they are now unreactive. So acyclic hydroxyaldehyde or open chain form, it still has the free, a free aldehyde and hydroxyl group. That's why somehow it's unstable. That's why trace amounts lang siya in equilibrium. Alright, so again, supplementary discussion. For your alpha D glucose, the methyl alcohol and your anomeric alcohol, they are trans to each other. Okay? So recall class, di ba? Ang methyl alcohol, andito po siya. Ayan, so that's why they are trans to each other. For beta, again, this is your methyl alcohol. They are cis. They are both above the plane of the ring. Okay, but again, guys, much easier to remember ang alpha baba and beta-as rather than this one. All right, so aside from intramolecular reaction, your functional groups present, your hydroxyl and carbonyl groups, are also capable of reacting to different reagents. So we listed here the relevant reactions that are known to appear in your board exam. So for the reactions of the hemiacetal functional group and your hydroxyl group, we have four. But we can divide this actually, class, into two main reactions. All right, so for the first two reactions, you can see here, if you will observe the structure of the products, only the hydroxyl group directly attached to the anomeric carbon were converted. Okay? It's the only functional group converted because we used relatively a mild reagent. For the first one, glycoside formation. So ether in nature ang glycoside class ha. So here, using your alcohol in an acid catalyzed reaction, we can convert your hydroxyl group directly attached to the anomeric carbon to its ether counterpart. So any R group po yan class, depending on the alcohol used. Now, if we use an amine in the second reaction, instead of a glycoside, we will convert your hydroxyl group to an N-glycoside. So class, take a look. Ang point of attachment is the nitrogen atom. Sir, talagang dalawa yung products for these first two reactions? Yes, because the reactions are not stereo-specific. Alright, so I hope that's clear. Now, let's proceed with the third and fourth reaction. So class, yung reagents na ginamit dito, first one is a silver oxide with your alkyl halide, and then this one is your acid chloride or acid anhydride in the presence of your base, pyridine. Okay? They are relatively strong. That's why, as you can see, lahat po ng hydroxyl groups in the structure of your monosaccharide will be converted. All right? So for number 3, it will be converted to ether. For number 4, it will be converted to ester. So I hope you are still familiar with the functional groups ether and ester. Again, si silver oxide and then si acid anhydride or si acid chloride are very strong reagents. That's why lahat ng hydroxyl groups are converted to ether and ester respectively. All right, now let's proceed to the last 4 reactions. Here, class, ang focus natin is your carbonyl functional group. Number 1 and 2 are just redox reaction. Okay? So kapag reduction ang reaction, we are just concerned with the aldehyde functional group. Remember, class, pwede pa kasing ma-reduce ang aldehyde functional group. However, si alcohol, using your sodium borohydride reagent, it cannot be reduced anymore. All right? That's why for our first reaction, si aldehyde functional group will be converted to your alcohol. And we call the product alditol, di ba? So here we can also use hydrogen in metal catalyst. All right? Number 2, ayan, oxidation naman. So of course, here we can obtain 2 different products depending on the strength of your oxidizing agent. Ayan, if we use mild oxidizing agent, we will only convert the aldehyde functional group. Okay? Because much easier pong ma-convert or ma-oxidize si aldehyde functional group. And then if we use a relatively stronger oxidizing agent, of course, we will also convert the remaining or the alcohol functional group at the end of your monosaccharide structure. Sir, bakit yung ibang hydroxyl group po hindi na-convert? Because it is not readily available in the solution. Okay? They prefer more